So the QR code is here. This will take you to the informational page for 1,000 fungal proteins. You'll see that QR code in a few more slides as well. But to now pivot to just describing some of the core capabilities associated with 1,000 fungals. One of them, as I mentioned, is our cell-free pipeline. This allows us to go from a target gene to having a protein fully expressed and scaled up for structural analysis or functional annotation, functional screening. And we can adjust the scale of the expression experiments to meet our particular experimental needs. So we have very, very small scale reactions that can occur at 25 microliter scale and don't require any purification and can be used for functional screening. We also have much larger 6 milliliter reactions that are useful for X-ray crystallization and other studies using structural approaches. So we really tailor the experiments to the overall need, but one of the key features here is that we've expressed pretty much every single proteins from every single aspect of the tree of life, from humans, mammalians, plants, bacteria, algae, archaea, and especially fungal proteins. And so we have multiple different cell lysates that we use cell-free expression for, but this is definitely a pipeline that comes into play whenever you only have a gene ID or protein ID or sequence. Cryo-EM is another one of our core capabilities. This allows us to get structural details of your protein without requiring large amounts of protein or even crystallization. It also allows us the ability to work across scales. So if you had a fungal protein that was largely uncharacterized, but you think it might be associated with a secondary metabolite, and that secondary metabolite has very limited structural information on it, if we were able to, or you were able to, isolate that particular metabolite, we could actually use small molecule microelectron diffraction to try and solve the structure of that secondary metabolite, and then link it with functional assays to your protein of interest to show a direct association and ascribe functional annotation to it. In terms of individual proteins or protein complexes, we can use both protein microelectron diffraction as well as single particle imaging. Single particle imaging is particularly nice because here you do not need to crystallize your sample. You can just start with a fairly pure sample of protein, put it into the TEM grid, put it on a TEM grid, freeze it so fast that the water can't crystallize, and you dynamically immobilize the samples, put it into the microscope and get images directly of it. That's what you see in the blue column. On top is a cryo-EM image where the black or darker densities are representative of the protein scattering on a water or buffer background. And below you can see an overall reconstruction. With this approach, without crystallization, you can still get 2 to 3 angstrom resolution in many samples. And then finally, if you had a cellular strain and wanted to look at the organization or compare differences of that fungal strain under different environments, to look at the localization or abundance of a particular large heterocomplex, you could do that with whole cell tomography. So cryo-EM is quite powerful that it allows you to analyze things across those scales. But another thing is pulling in native mass spectrometry and other complementary approaches. In this case, I'm going to show you an example where we coupled cell-free expression with cryo-EM and also native mass spec to address a scientific challenge. Here you can see two proteins, the monomer of PDX 1.2 and the monomer of PDX 1.3 in yellow and pink respectively. PDX 1.2 is a pseudoenzyme. It's non-active, whereas the PDX 1.3 is an active version of the protein. And prior to this work, the model was that the pseudoenzyme-enzyme interaction came through an association of two homomeric 6-mer assemblies interacting together. And so rather than having a fully intact and fully active structure of PDX 1.3 as the dodecameric barrel, all in pink on the right-hand side, you would modulate the overall activity by having these mixed complexes of these 6-mer rings. And doing self-re-expression is nice because we have the ability to actually tailor the amount of DNA that gets input for each of these particular clones. These are done co-translationally, so the two clones are added at the same time. We generate both the proteins at the same time, and then they can self-assemble. And by varying the amounts of them, we were able to adjust the stoichiometry, pulling in native mass spectrometry, where you don't actually degrade the sample, but you wind up being able to look at the fully intact sample with mass spectrometry. We're able to track that we can actually see every single stoichiometric assembly, which already with the self-re-expression with native page as well as the native mass spec, already identified that the 6-mer and 6-mer model probably isn't true. But then when you couple that with cryo-EM and look at the entire story and have the full ability to sample the entire stoichiometry between it, you can see that in reality, what's happening in the cells is that the PDX 1.2 and 1.3 are able to associate at any position within the 12-mer dodecameric ring structure and in any relative stoichiometry. And so it's a much more complex model for regulation. And we were able to illuminate that by combining all of these techniques here at EMSL. Now, expanding beyond just the single protein level, I mentioned that we also have the ability to look at cell strains. And so here you also have some optical approaches. We have many optical approaches at EMSL, including super resolution fluorescence, but CARS SRS is one that also allows you to track temporal dynamics, but do so in a label-free manner where you, if there is a spectral signature associated specifically with an organelle or a complex or overall strain of interest that you care about, you can track it with those signatures and see the overall change over time. And so you can see here, if you compare in the far right, the column of 1631 versus the column on the left, you can see that there's several cells that actually disappear in the 1631. These are cells that are losing the ability to do photosynthesis and so forth. The same type of work can be applied to fungal strains, even though this particular example was cyanobacteria. Another one of our capabilities, we don't have time to go into all of them, but another one that can be applied kind of if we're working with whole strains is nanopots. And this is a method that's been developed here at EMSL. It's considered nanodroplet processing in one pot for trace samples and can be used for metabolomics or proteomics. And I don't want to go into full detail here because this is current ongoing work, but it is an example that is working with fungi. In this case, they're working with Euroia, and they're using a Selenin-1 robot in order to isolate individual Euroia cells, deposit them into micro wells, and each one of those micro wells are isobarically labeled in order to be able to track them. And then they do this processing within these wells and then load through an auto-injector these very small nanoliter volumes for LC-MS analysis. And very excitingly, they've been able to start to identify metabolomic changes from these single cell experiments truly at the single cell level. So this is another exciting aspect of work that could be done within the 1,000 Fungal Proteins project, if interested. And so with that, the last aspect is a switch from experimental work more towards computational space. And so AlphaFold 2 had a huge splash over a year and a half ago. I'm sure everyone in attendance here has heard of it or is using it and so forth. So I don't really need to go into the full history and details, But AlphaFold 2 and other subsequent generations of similar methods are very powerful for in silico structure determination. And they've populated the protein data bank from having about 200,000 unique structures to now having over a couple 100 million unique structures associated with proteins. So AlphaFold 2 and machine learning for in silico structure determination has been a huge windfall. At the same time, we've also identified, not us, but others, have identified that there's still hundreds of millions of proteins that have low confidence in these models, or regions of low confidence. In addition, the publicly available repositories only include the pre-computed monomer models, not multimers, and they also don't provide direct analysis, direct accessibility to AlphaFill models, which allow you to actually identify potential ligand binding and other things. And so these are areas where EMSL does come into play. We have full workflows for AlphaFold 2 and other bioinformatic pipelines. And we intentionally, with every single sample that we wind up putting through the 1,000 fungal proteins pipeline, we intentionally do a screen with AlphaFold for potential oligomerization with multimer models of monomer, dimer, and everything up to about 12-mer, depending on the overall sequence of your target protein. And we use this to inform our experimental studies. So hopefully this has now given you kind of a brief overview of some of the core capabilities, not all of them associated with the 1,000 fungal proteins project, but these are largely the ones that are applied to most every sample that does come in. And so with that, I will turn it over to Irina to continue with the science examples.
Thank you, James. I'm just trying to get control of the slides. All right, now I'm in control. Good afternoon, everyone. I'm happy to see you here, and I will show you a few scientific examples that illustrate the application of those capabilities that James showed to you. So the first scientific example includes our previous collaboration work with Jiwei Zhang. So Dr. Zhang's Dr. Zhang's research is mainly focused on brown rot fungi. So they are the most important agents in degradation of food. So these organisms have evolved quite unique mechanism, which is using reactive oxygen species to initiate the breakdown of lignin cellulose. So he curated a list of genes, enzymes, which he found relevant to this process, but he had no means of validating or characterizing of their functions. So he had some predictions and so on, but he didn't have any access to the fungal proteins. And that's where we came in. So we generated a bunch of proteins for him. So we used our cell-free expression platform. And here on the slide, you see an example of gel showing cell-free expression screening screens that we generally do. So for example, the first two lanes show here a control reactions where we don't have any protein expression. The next two lanes, lane 3 and 4, shows the expression of gene 9701 fungal protein. As you can see, it's well expressed as a crude protein, but it's because it's a membrane protein, it remains mainly insoluble. To compensate for that, cell-free pipeline has an ability. You can add any additives that you like, I mean, to some degree. So for membrane proteins, we usually add nanodiscs, so we can maintain the proper folding environment and keep it soluble and such, and therefore functional. So you can see it in lanes 5 and 6, and the lanes 7 to 10 show you a few examples in the case of soluble proteins. So for his particular case, we target a range of soluble and membrane proteins. And those proteins, once we obtain them, we are subjected to different functional assays. And using this approach, we actually successfully characterized for key enzymes in reactive oxygen species pathway. And the results of this work are now being published actually last week in the Applied and Environmental Microbiology journal. So we're quite excited about this result. So we know that our system works for fungal proteins, but we also encounter a few critical things that we know that we need to add to our pipelines. For example, as you know, fungal proteins, a lot of them have different post-translational modifications. And because of that, we really need to make sure that we can provide those PTMs. So we are basically considering creating and adapting a fungal cell-free lysates in the future into our cell-free pipeline to make sure that we create really ideal protein folding environment. And eventually we want to do in vitro metabolic profiling as well. So that is similar to the previous approach. We used a similar strategy with another EMSL user, John Magnusson, who had a strain of Aspergillus. That particular strain exhibited an elevated production of cis-aconitic acid. So it's very valuable chemical that is attractive for its potential use in biodegradable polymers, surfactants, plasticizers, etc. So what he did previously, he compared global proteomics between wild type and this particular strain, which is lacking CAD8 gene. And he found a putative membrane protein, which his team suspected might be a transporter of this cis-aconitic acid. So to verify that this is indeed the transporter that they suspected, we again implemented a cell-free expression pipeline. We also express this protein in the form of the protoliposome because this is a membrane protein, so it needs to be encapsulated in the lipid environment. And we also developed a fluorogenic assay. So in a way that when this expressed protein is embedded in protoliposome, those liposomes encapsulate a fluorogenic dye, which is sensitive to pH. So when you have no acid in the buffer where they are in, you should not see any fluorescence. If your protoliposomes have suspended in a buffer which has an acid, and if that acid is being transported inside the protoliposome, you will see the fluorescence signal. And in the case of cis-aconitic acid, when we added it to the buffer, so the protein were able to transfer it in from outside the buffer inside the protoliposome, and this is what we see as a bright reddish spots. This is the protoliposome bodies themselves being light up as it's transferring the acid from buffer to its inside. We, of course, you want to have a lot of controls with that, and we substituted this cis-aconitic acid with very similar itaconic acid, and we can see an immediate drop in its transport efficiency, like 15-fold, so it's quite specific. We also tested a few other acids, such as 3-hydroxypropionic acid and citric acid, and as you can see, that basically the transport is non-existent. And to make sure this is not due to a loss of fluorophore efficiency over time, we again repeated these experiments with this aconitic acid, and we saw the transport again. So we were able, so with this particular approach, we were able to assign a function for this putative membrane protein transporter. So we know now for sure that it's this aconitic acid transporter, and we are now working on its atomic model. Here you can see the structure of it in AlphaFold 2. Just shifting a little bit from the functional annotation, we want to highlight our cryo-EM capabilities. So we have also used cryo-EM to begin structure characterizing fungal cellulosomes, and this was done in collaboration with Michelle Amali. Michelle Amali is a longtime user of EMSL and JGI, and she has been at the forefront of the discovery of fungal cellulosomes. We have paper now in review at Nature Microbiology, where we provide detailed biochemical analysis of these complexes. Specifically, what you see is the carbon nanofibers, and then you have like a little densities next to them, and this is actually your fungal cellulosome. So using cryo-EM tomography and the slices of which that you can see at the bottom, we can reconstruct those dark density and present you with a three-dimensional high resolution model. So it looks like actually like a man walking on a stick. Because of the nature of those complexes, they are being isolated from natural isolates. So it's like quite heterogeneous in terms of composition and architecture. So to use them as they are in like high resolution single particle cryo-EM is impossible. So basically what we're doing next now, we're doing cell-free co-expression to really co-express select protein components of this complex to have simplified and more defined samples that we can work with. Also, we initiated, as James mentioned, so we launched this JGI pilot project leveraging microcosm database. So this is a fantastic resource created by team at JGI. Here you can, so as I want to remind you, so they generated 142 gene families of unknown function. So we decided to focus on 14 initial targets. In the last week, we actually were able to confirm the expression of all of them. So this graphic is a little bit outdated. Half of them are already purified and frozen and actually are being queued into the cryo-EM screening session for imaging. So we very excited about the prospect of being able to fill in some gaps in this knowledge, and as well as expanding our collaboration with JGI towards to tackle additional families as well. In terms of how to access it and how to work with us, I want to highlight, first of all, even though the pilot project was including JGI gene families, our goal is to have the science is rather user-driven. So we want to hear what you need and what kind of science problems in fungal research you're trying to address. Maybe you already mined a lot of interesting gene targets that you want to study. And this is kind of science that we want to use as seed projects. We want to highlight that this is project not just for 1 year or 2 years, 3 years. This is a project that we are trying to pursue for the next decade. So we want to initiate, we need to know your science to have those seeds planted so we can further tackle really large grand challenges and tackle consortia-like projects. The best way to work with us is through EMSL user proposal types, proposals. I want to highlight that 1,000 fungal proteins is a high interest topic in the next, in this year and the next year. So we have to highlight it in the proposals. So you're gonna get a higher priority. We have 3 main large proposals, such as EMSL exploratory, large-scale research, and FICAS call. So FICAS call actually include collaboration with JGI, Argonne National Lab, and Oak Ridge National Lab. So you can request resources in all these user facilities through a single proposal. However, don't be scared if you really don't have enough science that you really want to pursue. You just maybe have a really small project or a few interesting targets that you identified and you're not ready to submit a large-scale proposal or anything like this, please feel join the effort, and it will be at no cost to you. Just follow this QR code, contact us, let us know that you have like a few targets that you're interested in working, and we can see what we can do. Again, I want to remind you that the minimal requirements that you need to have is really a target gene or protein sequence. So really, this particular web form, please contact us to get a link to it. But this is basically, you fill up all your information about your proteins, and we give you back, we will get back to you in a month, in a 1 month time frame, and tell you if it's possible that we can work on your project or not. So please limit to no more than 5 proteins or 5 target genes in mind for that. This is really like small scale. And just to highlight general considerations and minds that we usually have here when we're working with proteins, you don't have to really know exactly which cell-free system will be the most appropriate for you. This is something we have to worry on our side. We will identify the best system and approach for your targets. The minimal thing, what we want from you is UniProt entry ID. If you know it's membrane protein, soluble protein, its molecular weight, its properties. If you do know what kind of cofactors are needed, because this is necessary for cell-free supplementation, maybe these cofactors are needed for folding or for its activity and so on. So if you have this information, that would be useful. Again, if you have from previous analysis, you know that your protein target has some PTMs, please let us know. That will also guide our decision in terms of what kind of cell-free platform to use. This is assembly into larger homomeric and heteromeric complexes. So maybe from your protein interaction studies or something, you have suspicion that it's forming some kind of complexes with other protein split. Let us know, and we can try to run and in AlphaFold 2 and see if it's indeed the case, and maybe guide our workflow a little bit differently. Again, all these are optional. It's not required. Again, the minimal, if you just give us UniProt ID, that would do it. And again, if you have a purified sample, we will ask you very standard questions such as concentration and buffer and native state. Just want to highlight one more time about our main cell-free expression pipeline. I would say this is based on the wheat germ lysates. It's a main workhorse on our side. Again, as James mentioned, it's amenable to for the expression of proteins of different origin, bacterial, fungal, algae, plant, and human gene. The clones don't have to be optimized or anything like this. So if you have those, you can ship those to us. I want to highlight that main reaction are usually conducted on robotic synthesizer, and we get very high purity. So I would say for the majority of targets, we get the purity higher than 90%. And of course, depending on the needs, we will usually have 2 main scale-up formats, usually Maxi for structural biology application, where we can do up to 6 proteins. And for functional annotation, we can do in 24L format synthesis. And because of all this previous work and now next steps, EMSL decided that the next summer school going to be devoted to this 1,000 fungal protein projects. This summer school will provide 25 attendees with 5-day hands-on experience. So every single attendee will be able to select a protein of choice that they want to work with, so they can express, they can do cryo-EM screening, AlphaFold 2 model prediction, and so on. So please save the date. It's going to happen on July 22nd to July 26th. please use this QR code to register, but please keep in mind we have only probably 1 week left before the registration closes. So please hurry if you want to attend. The main topics for this workshop is going to be cell-free expression, cryo-EM screening and data collection, AlphaFold 2 and data image processing, optical Raman imaging and cellular infrastructure in nanopods. So it's quite a wide variety of topic, and it's going to be a good experience for your students, postdoc, or if you want to visit yourself as a PI of any grant, we are happy to have you here. And if you do want to learn more about those capabilities in greater detail, please visit EMSL YouTube channel and EMSL Learn website. So every capability that we've mentioned is being described, and there is much more examples over there you can find, including how to submit the successful proposals. But we generally advise, please reach out to us if you don't know. We are here for you to help you. So as at the end, I would like to thank you everyone for your attention, and I would like to thank you EMSL for funding and launching this 1,000 fungal projects. Please reach out to us. We are happy and we're here to answer all your questions, and please visit our website using QR code. So thank you so much. I think we are ready for questions.
Yes, hi. Well, thank you, James and Irina. That was fantastic. And so we are going to now address some of the questions that we've received in the chat. So I guess we can start off with, so Lenny had asked or had mentioned in the chat that we are a team of data computing scientists with zero to little biochemistry background. Could you please comment on what are the data computing challenges found in the 1,000 Fungal Proteins project?
I could take a stab at that.
Yeah.
So I guess 2, maybe 3 things come to mind. First, AlphaFold and in silico modeling generally has a limit to the size of the protein, or the number of amino acids rather, that can be modeled. And this is largely dependent on available GPU hardware. So even with the best hardware that's available to us here at EMSL, we're limited to somewhere around 5,000 amino acids as the max number that can be modeled. And so if you have a very small protein, you can do a multimer model of 24 or even more copies and so forth without a problem. If you have a protein that's 1,000 amino acids long, you can't get past modeling a pentamer. And so in real life, it may exist as a decamer or dodecamer or something much larger. And so we couldn't actually sample that multimer space right now just because of the limitations on overall modeling. So finding ways to accelerate it or minimize the limitations there would be one active area that could help. A second is that computational screening for possible ligand binding is a very large phase space with very large libraries of possible ligands. So accelerating that is good. And then I guess the last part, which is of active research by many people in the field, is recognizing that AlphaFold still has limitations with membrane proteins, especially modeling segments that would be within the lipid bilayer or pass through, and also intrinsically disordered proteins. Those are the 2 main classes of proteins that have very low confidence regions with them with AlphaFold. So those would probably be the data challenges that come to mind.
Fantastic. All right. Our next question is from Armando, who asks, are there limitations on working with certain types of proteins, like membrane proteins or proteins with intrinsically disordered domains?
I think I can take with that the question. Usually usually we express all the proteins. We never have any issues with the expression. But then when you go to activities, we don't know, did we express a functional protein or not? Our approaches in terms of, so it has to be validated by a set, did we get a functional protein or not? The main problem generally that we find with putative proteins and their predicted functions is not that we didn't produce a functional protein, it's just we don't have the right assay in hand. We don't know what is the right substrate for it or something like this. So it's very difficult to judge, especially for these unknown proteins. For membrane proteins, we have really pretty straightforward workflows. We usually embed them in nanodisk or liposomes, so that has never been a problem. And I think the end of it, Linda, what was the last one?
Oh, the last question, intrinsically disordered domains.
So we currently have a few ongoing EMSL user projects with such candidates, and we find that with one particular, we actually have very good co-expression results. We expected to form a multimer, and we do observe multimer formation, so it's a good indication for us that it's performed as expected. These studies are currently ongoing, but they look promising to us. And they are fungal proteins with disordered regions.
Okay. Another question, what type of PTMs can you detect? Are you able to look at less common PTMs, such as nitrylation, chrotinylation, thiolation, etc.?
Yeah, so the EMSL 1000 proteins project is pretty narrowly focused and so forth. So I would say that the majority of samples that come through the 1000 Fungals direct pipeline will be investigating pretty common and pretty easy to detect PTMs, largely things that can be detected with native mass spectrometry, so presence of potential glycosylation and phosphorylation and other things. Those are going to be the main targets. If you need more specific ones or more of the complicated to detect ones, those would be things that would still be recommended to go through a general EMSL call. They have access to more complex methodologies, better equipment, and more expert staff in that area.
And speaking of proposal calls, we have another question. If I have a large project with many targets of interest, can I still apply directly to the EMSL 1000 Fungal Proteins project, or do I need to wait for the next round of EMSL proposal calls to open?
I would say definitely apply. You would be limited to somewhere probably around 3 to 5 targets. So then you could choose your highest priority targets from your larger list, or maybe the most diverse targets. And then we would initiate work on that, and then any results that you got from the more direct access could then be leveraged or used as preliminary results when you put in the future larger scale EMSL proposal. So don't feel like you have to wait. If you can find a smaller subset that could have the work initiated on, that's definitely of interest.
Okay. We have another question from Lenny. I don't have a background in the fields of biology, biochemistry, biophysics, chemistry. I am not a student or a postdoc anymore, but could I still apply for summer school?
So the intent of summer school was to kind of train the next generation and provide a mechanism to get more users and more groups kind of engaged. I don't think that there's a restriction exactly on overall career level and so forth. The one thing that I will have to say is that there is a restriction on total number of seats. And so I think one of the determining characteristics will be the ability to potentially leverage access in the future to accelerate research and overall productivity towards characterizing fungal proteins. So I think as long as your application describes how you would be able to fit in that phase space and why it would be particularly beneficial and impactful for you, then it would go under consideration.
Great. So the next question, what is the likely time frame for solving a protein structure? If I already have purified the protein, or if we use the cell-free pipeline?
I can answer it again. So short answer would be every protein behaves differently. So can't really ascribe a single time frame. If you have a well-behaving and biochemically pure sample that you're providing us, then a structure could be solved in about 2 to 4 weeks after receipt. Many proteins, however, aren't very well behaving, so it can take several months or longer. Some of that is dead time of us having the sample in the queue to get on the microscopes and so forth. But the need to potentially screen multiple additives or stabilizing factors to try and assist and get the structure to be amenable to structural analysis can impact the time frame. In terms of using the cell-free pipeline, it's an interesting question because we may have a little bit more flexibility. Because we have 3 different lysates, we can try to express in all 3 different types, bacterial, mammalian, and wheat germ. And again, every protein is different, but we have several examples now of solving a structure within only 18 hours after initiating a cell-free expression. If we can use the bacterial cell-free lysate and the protein expresses and folds natively. So very fast turnaround there. But not all proteins, especially fungal proteins, can be done in the bacterial lysate. For wheat germ expression system, our best case to date has been 9 days after receiving the gene clone. We've had a fully purified, fully expressed, purified, and solved structure to 2.5 angstroms. And interestingly, it took 27 days, so 3 times longer, just to simply receive the gene clone. So 9 days turnaround there. But as I mentioned, every protein is different. So we have other proteins using those same pipelines that are still trying to get clean structures even after months of effort. So every protein is different and you kind of start back at 0 with most of them.
Yeah. We have 5 minutes left. And I see one question that's still out there. So I'm going to ask this question, but audience members, if you have anything else, please let us know. Drop your question in the chat. So the last question I have so far is, do I need to already have an idea of function for the protein to go through the functional annotation workflow?
I would say our main bottleneck is functional assay, because if you want to characterize protein, at least you need to have function, you need to have an idea what it might be. We don't have any, we can synthesize the protein for you, we can purify it, we can solve the structure, potentially we can set up certain types of fluorophore type style assays or something like this, but we have to have some kind of knowledge, because for each assay you have to know substrates, you know what you're detecting, right? So some kind of prediction of the function, predicted function, you have to know. In order to learn for completely unknown protein, you have to set up certain kind of small molecule screening assays or something like this. We don't have capabilities like this here to do such things. I don't know, James, do you have something else in mind to add?
Yeah, I would potentially add that in the case where you don't know function already or have an idea, we are able to partner with some of the computational folks here at PNNL in order to do some library screens computationally to see if it can identify potential ligands, and that might inform function. But that requires extra resources and time and so forth. And so the only other method that we can really apply besides that potential computational screening would be to actually solve the structure of the protein and then try to do a scan based off of the protein fold that gets identified to see if we can identify other common folds and then identify the function there. So if you already know or have an idea that would bracket kind of the type of functional assays that would be needed in order to validate function, then you have a much faster path to functional annotation with our workflows. If not, it's not impossible, but it definitely engages other parts of the workflow that will take longer.
All right, well, I think this is it. We don't have any other questions, and we are just a couple minutes from the hour. So with that, I will say adieu to everyone. Thank you for joining us. We hope that if you know of anyone who would be, any students or postdocs who would be interested in participating in the 1000 Fungal Proteins Summer School, we encourage them to apply. Applications are due by April 17th, so there's still about a week left for them to get in an application. If you have any questions, you're more than welcome to contact James or Irina, and we can leave their email addresses in the chat if you haven't already written them down throughout the webinar. And with that, thank you. Happy Wednesday, everyone.
Goodbye.
Bye-bye.
Bye.